plant anatomy. First topic is meristematic tissue. Meristematic tissue mean embryonal cells or juvenile cells participated in the periclinal divisions or mitotic divisions that develop into complete plant body. Generally, the meristematic tissues are developed from the embryo. All plants of embryos are contains only meristematic tissue. The meristematic tissues are in diploid condition. The diploid condition meristematic tissues are embryonal cells or juvenile cells participated in the periclinal divisions or mitotic divisions. They develop into complete plant body like root system, shoot system. See, meristematic tissues are present in the plant body like in the terminal parts of shoot apex, axillary parts of node, and the terminal part of root. Mainly three portions are having meristematic tissues. The meristematic tissues are having only one function, that is division. Always these areas of meristematic tissues participated in the division, they develop into complete plant body. The meristematic tissues are having triangle, rectangular, hexagonal, polygonal, or irregular in shape. In this diagram, the meristematic tissues are in hexagonal in structure. Each meristematic cell containing proper cell wall. The cell wall is made with the cellulose and hemicellulose chemical. It is in elastic nature. Below the cell wall, a thin plasma layer is present. Below the plasma layer, we observed one large nucleus. If meristematic cells identified with only one large nucleus, it presented the center region. But in adult cells, the nucleus is small in size. It present in any corner of the cell, not in the center region. But in meristematic cell, large nucleus is present. The large nucleus present at the center region. Around these nucleus, large nucleus, liquid cytoplasm is present. In this liquid cytoplasm, mitochondria, remaining all cell organelles are present except chloroplast. See, around these large nucleus, more number of small vacuoles are present. It is another identification of meristematic cells. Meristematic cells contains more number of small size of vacuoles. They are present at the large nucleus. They are present around the large nucleus. But in adult cells, adult plant cells, only one large vacuole is present at the center region. First point is large nucleus. Second point is small, more number of vacuoles are present in the meristematic cells. And all meristematic cells are compactly arranged without any intercellular spaces. These are having only one function, that is division. They didn't take any rest. They are continuously participated in the division. They develop into complete plant body like root, shoot, leaf, flowers also. The meristematic tissues are in three types. They are apical meristem, second one intercalary meristem, third one lateral meristem. Generally, in all plants, these are the three types of meristematic tissues are present in the plant body. See, we observed in this diagram shoot part. The shoot part contains apical meristem, intercalary meristem, lateral meristem. Apical meristem present at the terminal part of shoot. The terminal part of shoot of meristem continuously participated in the division. They develop into more number of tissues. The tissues are helps in enlargement of shoot part, length of shoot part. It mainly helps in increasing of length of shoot part on the terminal part of shoot. At the node area, intercalary meristem is present. The intercalary meristem present at the node area of the shoot. These are participated in the periclinal divisions. They are developed into more number of tissues. The more number of tissues are helps in width increasement of a node, increasing of width area by the development or division of intercalary meristem. And between the two nodes, here it is from node area, here it is another node area. Between the two node areas, internode part is present. In this internode part, lateral meristem is present. The lateral meristem also participated in the periclinal divisions. It developed into increasing of size in the internode area. These are the three main important meristem tissues present in this shoot. At the same time, the apical meristem and the lateral meristem also present in the root part. Terminal part of root containing apical meristem. It participated in the periclinal divisions. It helps in length of root system, and the lateral meristem also present in the root system. They are participated in the periclinal divisions. They are helps in the enlargement of the root area. And from these meristem cells, root apical cell theories, shoot apical cell theories are explained by different scientists. See, first we discussed about the root apical cell theories. Terminal part of root contains meristem. That meristem is called apical meristem. The root apical meristem or apical meristematic cells, how to develop into complete of root. The theories are explained. First theory is apical cell theory. It proposed by Hofmeister scientist. According to Hofmeister scientist, on the terminal part of root, only single tetrahedral apical cell is present. The single tetrahedral apical cell look like a pyramid in structure. It has 4 faces. The 4 faces of pyramid cell or tetrahedral apical cell participated in the periclinal divisions. It developed into more number of meristematic tissues. The more number of meristematic tissues are developed into mainly 4 major groups of layers. One is dermatogen, second one is peribulum, third one is plerome, fourth one is calyptrogen. First one is dermatogen, second one is peribulum, third one is plerome, fourth one is calyptrogen. Dermatogen develop into dermis, epidermis of root. Peribulum develop into cortex of root. Plerome develop into stele of root. And fourth point is calyptrogen. Calyptrogen develop into root cap. It accepted in only pteridophyta plants. On the terminal part of root of pteridophyta, single tetrahedral apical cell is identified. So the apical cell theory is applicable for only pteridophyta plants. It is not applicable for the gymnosperms, not applicable for the angiosperms. And the next theory is histogen theory. Histogen theory proposed by Hans Steen scientist. According to Hunstein scientist, on the terminal part of root system, meristematic tissues are present. The meristematic tissues are called histogen tissues. Histogen tissues mean meristematic tissues. Hunstein explained all Tracheophyta root terminal parts. In Pteridophyta, gymnosperms, dicotyledons, and monocotyledon, angiosperms of monocotyledon. In Pteridophyta terminal part of root, single tetrahedral apical cell is present, that is histogen cell. Histogen single cell is present. It is in pyramid structure. It contains 4 phases. The 4 phases are participated in the periclinal divisions. It develop into 4 major groups of the meristematic tissues. They are calyptrogen, dermatogen, peribulum, plerome. Calyptrogen develop into root cap. Dermatogen develop into epidermis of root. Peribulum develop into cortex of root. Plerome develop into stele of root. These 4 major parts are developed from only single histogen cell. It is applicable for Pteridophyta root system. And this theory is similar to Hofmeister apical cell theory. And the second plant is gymnosperms. On the terminal parts of the gymnosperms of roots, 2 tiers of meristematic tissues, 2 tiers of histogen tissues are present. Lower tier of histogen tissues participated in the periclinal divisions. They develop into root cap and root epidermis. And upper histogen tissues develop into peribulum and plerome. Peribulum develop into cortex of root. Plerome develop into stele of root. And these 2 types of histogen tissues are identified in gymnosperms of root system, terminal parts. And in angiosperms, mainly 2 types of plants are present. One is dicotyledons, second one is monocotyledons. In dicotyledons of terminal parts of roots, 3 tiers of histogen tissues are present. The 3 tiers of histogen tissues. Lower tier of histogen tissues are participated in the periclinal divisions or mitotic divisions. They develop into epidermis of root called dermatogen. It develop into epidermis of root, and it develop into calyptrogen. Calyptrogen also develop into root cap. In dicotyledon, dermatogen, calyptrogen, both tissues, both layers are developed from the lower histogen tissues. And above these tier of histogen tissues, second tier of histogen tissues are present. The second tier of histogen tissues participated in periclinal divisions. They develop into peribulum of cortex. Peribulum develop into cortex of root, and upper histogen tissues develop into plerome. The plerome develop into stele of root. And these 3 tiers of histogen tissues are identified in the terminal part of dicot root tiers. And last one is monocot root terminal part. In this monocot root terminal part, 4 tiers of histogen tissues are present. Lower tier of histogen tissues develop into calyptrogen. The calyptrogen develop into root cap. Second tier of histogen tissues develop into dermatogen. Dermatogen develop into epidermis of root. And third tier of histogen tissues develop into peribulum. The peribulum develop into cortex of root. And fourth tier of histogen tissues develop into plerome. Plerome develop into stele of root. The histogen theory or Hunstein theory applicable for all Tracheophyta plants, and it is accepted by all botanists. The next theory is Carper-Cuppy theory. Carper-Cuppy theory also explained in the root apical cell theories. Carper-Cuppy theory proposed by Shep scientist. According to this theory, on the terminal part of root apex, initially only one single cell is present. The single cell is called central cell. The central cell participated in the periclinal or mitotic divisions. By these mitotic divisions or periclinal divisions, two types of tissues are developed. The two types of tissues are called Carper tissues and Cuppy tissues. Carper tissues are present at the center region of root system. They are called Carper meristem, and Cuppy meristems are present around the Carper meristematic tissues. The Cuppy meristematic tissues act as a cover of root. The Cuppy meristem tissues are participated in the periclinal divisions. They develop into dermis and calyptrogen. Calyptrogen is developed into root cap and dermatogen develop into root epidermis. And Carper tissues are participated in the periclinal divisions. They develop into periplum and plerome. These are the Carper-Cuppy theories and Hunstein theories and apical cell theories. These three theories are explained about the root apical cell theories. From these three theories, important theory is Hunstein theory, and it is accepted theory in all Tracheophyta plants. The next one is shoot apex theories. Shoot apex theories are shoot apical cell theories. The shoot apical cell theories are explained in different ways by the different scientists. The first theory is apical cell theory by Hofmeister. It's similar to the root apical cell theory. It is also proposed by Hofmeister scientists. According to this theory, on the terminal part of shoot apex, one tetrahedral apical cell or pyramid cell is present. It contains 4 phases. The 4 phases are participated in the periclinal divisions or mitotic divisions. They develop into 3 major groups of meristems. They are dermatogen, periplum, and plerome. Dermatogen develop into epidermis of shoot. Periplum develop into cortex of shoot. Plerome develop into stele of shoot. And here it is one important point. Apical cell theory is applicable for only pteridophyta plants of shoot, because in the terminal part of pteridophyta shoots, single tetrahedral apical cell is identified, but in gymnosperms, angiosperms, meristematic tissues are present, not single cell. So apical cell theory is not accepted in the gymnosperms as well as in the angiosperms of the shoot theories. It is applicable for only pteridophyta shoot apex. The next theory is tunica carpus theory. Tunica carpus theory proposed by Schmidt scientist. According to this theory, or according to Schmidt scientist theory, on the terminal part of shoot apex, only 2 types of meristematic tissues are present. They are tunica meristematic tissues and Carper meristematic tissues. From these 2 meristematic tissues, tunica meristematic tissues are present on the terminal part of shoot, and below the tunica meristem, Carpus meristem tissues are present. Tunica meristematic tissues are participated in the periclinal divisions. They develop into dermatogen. Dermatogen develop into epidermis of shoot. Below the tunica meristem, Carpus tissues are present. They are participated in the periclinal or mitotic divisions. They develop into periplum and plerome tissues. Periplum develop into cortex of shoot. Plerome develop into stele of shoot. And the next theory is in shoot apical cell theories, mantle and core theory. Montiel and core theory proposed by Popham and Chan scientists. According to this theory, on the terminal part of shoot apex, only two types of meristematic tissues are present. They are montiel and core. Montiel present at the terminal part of shoot apex, and below the montiel, core meristematic tissues are present. Montiel meristematic tissues are participated in the longitudinal division, periclinal divisions, and it develop into epidermis of shoot, and core tissues participated in the periclinal divisions or mitotic divisions, develop into peribulum and plerome. Peribulum develop into cortex of shoot, plerome develop into stele of shoot. This theory is similar to tunica carpus theory. Here it is, montiel is called tunica, core is called corpus. Montiel and core theory is similar to tunica carpus theory. And the next theory is French theory. French theory is proposed by Bouvard scientist. Bouvard scientist belongs to French country. The Bouvard explained meristem de-attenti theory. Meristem de-attenti theory. According to Bouvard scientist theory, on the terminal part of shoot apex, mainly four types of meristematic tissues are present. They are anu initial meristem, meristem de-attenti, pro-meristem, meristem medullary. From these four types of meristems, meristem de-attenti tissues are take rest position up to development of reproductive organs in the plant body. But remaining of meristems like anu initial meristem participated in the periclinal divisions, it develop into dermis, dermatogen, develop into epidermis of shoot. Pro-meristem, it present below the meristem de-attenti. It participated in the periclinal divisions. It develop into peribulum. Peribulum develop into cortex of shoot. And below the pro-meristem, meristem medullary tissues are present. Meristem medullary tissues are participated in the periclinal divisions or mitotic divisions. They are developed into plerome. Plerome develop into stele of shoot. From this total theory, meristem de-attenti tissues are take rest at engage of plant body. They are participated in the division at adult age of plant body, and they are developed into reproductive organs of plant. But this theory is not accepted because meristem means meristem continuously participated in the division. It didn't take any rest in the plant body. But Bouvard explained the meristem, resting meristem is present on the terminal part of shoot. The resting meristem is called meristem de-attenti theory, meristem de-attenti tissues. And the next important theory in shoot apical cell theory is Newman theory. It is accepted in all tracheophyta plants of shoot apical cell theories. According to Newman scientists, on the terminal part of all tracheophyta shoots, meristem tissues are present. The meristem tissues are called plex tissues. The plex tissues are also called meristem. In pteridophyta, on the terminal part of shoot apex, single tetrahedral monoplex cell is present. The monoplex cell is in pyramid structure. It has four phases. The four phases are participated in the periclinal divisions. They develop into three major portions. They are dermatogen, peribulum, plerome. Peribulum, plerome. Dermatogen develop into epidermis. Peribulum develop into cortex. Plerome develop into stele of shoot. And this theory is similar to apical cell theory proposed by Hofmeister scientist, and it is identified in pteridophyta plants. And the next one is simplex. Simplex theory belongs to gymnosperms. On the terminal part of gymnosperm shoot apical cell, terminal part of shoot apex, simplex mean single tier of meristematic cells. Single row of plex tissues are present called simplex. From this single row of meristematic cells or single row of simplex cells, dermatogen, peribulum, plerome tissues are developed by the periclinal divisions. They are developed into epidermis, cortex, and stele. And in angiosperms, two tiers of plex tissues or more than two tiers of plex tissues are present. They are called duplex. The duplex tissues are applicable for angiosperms terminal part of shoots. In dicotyledon, on the terminal part of shoot apex, two tiers of plex tissues are present. They are called duplex. From this duplex, lower tier of plex tissues participated in the periclinal divisions. They develop into peribulum and plerome. Peribulum develop into cortex of shoot. Plerome develop into stele of shoot. And upper tier of plex tissues participated in the periclinal divisions. They develop into dermatogen. Dermatogen develop into epidermis of shoot. And the last one is monocotyledon shoot apex. Monocotyledon shoot terminal part contain more than two tiers of plex tissues. It is also belongs to duplex. In this duplex of monocotyledon, three tiers of plex tissues are present. From each tier, main part is developed. Lower tier of plex tissues participated in the periclinal divisions. They develop into dermatogen, lower tier of plex tissues participated in the periclinal divisions. It will develop into plerome. Plerome develop into cortex of, stele of shoot. And second tier of plex tissues. Second tier of plex tissues participated in the periclinal divisions. It will develop into peribulum. The peribulum develop into cortex of shoot. And upper tier of plex tissues participated in the periclinal division. It will develop into dermatogen. From these three tiers of plex, upper tier of plex tissues develop into dermatogen. And next, second tier of plex tissue develop into peribulum. Lower tier of plex tissues develop into plerome. Plerome develop into stele. Peribulum develop into cortex. And dermatogen develop into shoot of epidermis. These four theories mean monoplex, simplex, duplex is applicable for all tracheophyta plant shoot terminal parts.